So let's welcome our next speaker, Lolitika Mandal. She Drosophila.
Okay, very good morning. Thanks so much to Amitav and Jonaki for having me here. It's a pleasure. It's my first visit to IIT Kanpur. Thank you. And I'm looking forward for more interaction. Please stop me wherever you want. All right? Thanks to our chairperson because he told us this should be interactive and that took the whole stress away from me. So it should be interactive, okay? So today I'm going to introduce you to my favorite organism, which is none other than Drosophila, and I lovingly call it as a Cinderella in genetics. And I'm sure you guys know who is Cinderella. And that Cinderella had her magic moment till 12 o'clock at night. But my animals, Drosophila, has her Cinderella moment forever. And today I hope that I'll take you through some of the classic examples which will make you believe that I'm not daydreaming. All right. So thousands of papers for the last 100 years. Thousands of research scientists across the globe investing time on this vinegar fly or fruit fly, who is an unwelcomed guest during the summer month on the table, your dining table, where there are a bunch of rotten food sitting there, and you see and you take a newspaper and you smash them, right? So this animal, fruit fly, is a subject of hot research. Why? That's what we are going to discuss today. Nonetheless, we all know this guy is instrumental in getting 6 Nobel Prizes for physiology or medicine, therefore a hot model system for 10 big scientists who made groundbreaking research in Drosophila. A little history onto it. It actually originated somewhere in the tropical region in Africa. Then it became cosmopolitan following the human population all throughout the globe. It was introduced to USA somewhere in early 90s. Some of the features that makes it unique is that the whole population of London can be stuffed up in few trays in your laboratory. That means they're extremely small and a great population can be housed in one incubator. More than that, it can be easily grown. It's a pocket friendly, of course. And what makes our life very easy is that the grandparents can see the grandchild within 45 days. Speeds of research life, right? So short life cycle, number of grandchildren, huge number of progeny, your N value equivalent to 500. So you have 3 trials, 3 pilot experiment, 300 samples, and you are done with reviewers' comments. So then this guy made its entry into research, which is sometimes in 1906, when at that time, the excitement in biology was people were debating onto the origin of species, which Darwin proposed. There was bombardment, there was discussion, there was confusion, and there was disagreement. All right, this will work. Along that time, this is Thomas Hunt Morgan, who decided to test the theories proposed by Darwin. And the organism that he picked up is none other than the Drosophila. So Drosophila made a smart entry in Columbia in 1906 as the preferred model of Thomas Hunt Morgan. Thomas Hunt Morgan, using all the facility that I rattled off to you about and good things about Drosophila, started the culture in innumerable number. Then one fine morning, one of the lab helpers, at that time he was just a work study student, later on became the PhD student of Morgan, none other than C.V. Bridges. He was washing a dirty bottle and he chanced upon a fly which was different from rest of the gang, which had red eye, but this guy has white eye. All right, so this fly caught the attention of both Morgan as well as Bridges. And you guess what? This actually changed the entire, it was a paradigm shift in our understanding of biology. Because it was a mutation. Morgan and his gang found out that it is due to a mutation in a gene, and they showed that this gene can be inherited, and that gene is located in a chromosome. All of it brought together or linked up several floating information into a nice little realization, which is these are all the raw materials of evolution. The first in vivo demonstration of Darwin's propositions. And that together also initiated a new discipline, which is none other than genetics. Few more years to go, we have discoveries that followed demonstrated that genes are situated, of course, on chromosomes, and this was the first chromosome map published in 1913. Later on, people could show through genetics that these genes can interact with each other, can influence with each other, are epistatic to each other. So gene-gene interaction, gene-gene crosstalk were all demonstrated using this vinegar fly. I'll jump few years from then, and I'll show you that slowly people started realizing that there is also fun in looking at other stages of development. And when they looked at certain cases, certain situation when the fly never emerged, so kind of cartoon like RIP, and you won't believe that this realization that we can visualize the effect of a gene on the survivability of an animal gave rise to a new concept, and that concept I want to demonstrate today. Drosophila genetics, during that time genetic code was explained, and empowered by gene editing helped us to understand not only that proteins are produced and this protein helps in biological function, it also helped us to realize that a mutation when happens to it, how it abrogates or attenuates the protein function, and how that protein function upon attenuation affect the biological function. This I will demonstrate using this, this help of these three marvelous people who got a Nobel Prize in 1995 working on the genetic control of early embryonic development. They are Ed Lewis, Christian and Aslin Volhard, and Eric Wishaws. Three of them invested their life in understanding how the unseemingly unpatterned Drosophila embryo suddenly gets patterned into different discrete structure in adulthood, like the head, thorax, abdomen, wings, legs, halter, right? You may ask me a question right now, how does it get related to this finding to Drosophila, this finding to human being? What exactly I want to show you that the genes that they described in fly was later found in mouse, then related in man, and then a mutation in man was shown to be responsible for cancer. Among the cabinet of this huge amount of genes that they had found out, I'll take just one example to illustrate this phenomena. What the approach they had taken is they fed the flies with a cancerous substance, and then they screened the embryos for a phenotype that seemed seemingly different from the normal. So normal phenotype is where you have naked cuticle as well as few ridges that you see. So let us keep it very simple. The tentacle should be ornated with the whole epidermis should be ornated with denticle and naked patches, and this structure enables the maggot to move around, right? So now what they did is they screened for embryos that had different or difference or disorganized denticular pattern, and some of them even didn't have head, they have two tails. They lost both their ends, that is they neither had head or neither had tail, or some that totally lacked all this denticular structure. Among all this mutant, they caught attention of a particular mutant which was covered up by these denticles. There was not a little naked patch left, and they called it hedgehog. And you know it is because hedgehog is a desert, what? It's a desert animal with lots of spines on it, right? Drosophila mutants are actually named upon the function, normal function done by that gene. Suppose this hedgehog gene is there, then it should have been a normal looking embryo, but since the gene is abrogated or attenuated, you get a hedgehog like phenotype, right? So the name is actually the mutant's description, all right? I used to be very happy during my masters because whether I know the gene function or not, I could predict it from the name of the gene, like wingless. Oh well, it must be responsible to do something with the wing, right? They went ahead and found that this gene, where this gene is actually located, and then one step further, what the normal function of this gene. they understood that using, of course, the sophisticated Drosophila genetics, they could know that these few genes, these few cells on the epidermis that expresses hedgehog actually secretes a signal that patterns the epidermis on the both side. They also realized that the function of hedgehog is not limited to embryo. Hedgehog is also required to pattern the posterior half of the Drosophila wing. All right? Now, what will happen if I now force hedgehog to appear somewhere here? You see, excess of hedgehog will cause an extra wing. Now, what is Lolitika doing here? What is this result being so exciting to her? Right? The excitement comes in when you realize that this phenotype can be explained when you look at defects in human, right? When you have an extra thumb or extra pinky, which is polydactyly, right? And what was amazing is that this polydactyly is also caused by a gene which is a near about relative of hedgehog, none other than sonic hedgehog. Now, if you look at the expression pattern of sonic hedgehog, it is beautifully present in the limb bud, and it doesn't need any introduction in this audience with Amitav and Amitabh's students around, that hedgehog expresses in the posterior part of the developing limb, and it is required for patterning the posterior half. Now, if I do the same experiment that I did in Drosophila, put hedgehog in a wrong position, what will happen? You will have extra digits. Now, do you understand why I get excited that this mechanism of having extra digits, the significance, the cocktail, the transcript that were required for normal digit formation was actually first worked out in using Drosophila wing as the model. And this two gene, which is hedgehog in Drosophila and sonic hedgehog in the vertebrate counterpart, comes and amalgamates at that position, where you can't do this kind of experiment straight away in vertebrate, but you can solve all the riddles in a smaller organism like Drosophila and then try to answer a question pertinent to your vertebrate development. And in this regard, I would actually thank Subbu for making a fantastic introduction about what is the significance of a model system. The role of hedgehog doesn't stop here. People soon found that this hedgehog gene, when becomes abnormal, causes overproliferation of cells, skin cells, and that results in cancer. And therefore, you see, that led to the brainstorming session of creating a drug against cancer. People found a way to block or reduce hedgehog function, and this is the greatest advance in cancer therapy because a pill does exist which can, independent of chemotherapy, can be administered to the cancer patients. So what have I told you? I told you that Drosophila shares body pattern. The genes that are responsible for patterning of Drosophila also are found to be useful for patterning the human body. 100 years of research till now have shown that the organ systems and the nervous connection, or how we breathe, or how our blood cells move around in our body, can also be reflected in Drosophila that has similarity with the vertebrate counterpart. So what do? We used fly research for stem cell biology. We use fly research for sleeping. That's the last Nobel that we had. We also use fly research to understand jet lag. We use fly research to understand addiction. ADH gene, alcoholism, was first worked out in Drosophila. We understand memory using Drosophila. We also understand age-related defects, especially Huntington and neurodegenerations like Parkinson, using Drosophila as a model. Drosophila model first gave the gene Parkin, was first cloned in Drosophila. And then you know this too, that Drosophila has been sent as the fruit fly astronaut in the space to understand behavioral changes. And this is not my endeavor, it is NASA's endeavor. So, recent interest, if you try to map, Drosophila has invaded every aspects of our research, right? I'm not claiming right here that this is the best possible model, but I'm claiming here it is the testable genetic model that can give you a good understanding of a process that is going on during the development as well as disease pathology. What are the tools and techniques? Tools and techniques, well, we have a huge resource. Drosophila community is actually enriched by the resources of fly lines, RNAi lines, overexpression lines. They are all available, and the community is very sharing. So we often get gifts without throwing in money. All right. But the genetic tools, I didn't introduce you to the life cycle. So there is your adult fly. This fertilized egg takes 1 day to become a first instar. This guy takes 1 day to move into second instar. This takes 1 other day to move into third instar. And third instar stays there at about 2 days, then it moves into metamorphosis, where hell a lot of remodeling takes place, and out comes a new adult which is ready for mating within 8 hours of emergence. Now, if a gene function at different times is to do different things, Drosophila has an answer. Why? Because then you can use temperature sensitive mutants to attenuate the function at that time. For example, I know that there is a gene X which is required throughout the development, and suddenly I see that gene X expresses in the developing eye of Drosophila, right? So now if I switch off gene X from very beginning, I might not get a viable individual. It's not smart to do that experiment. What is smart? That you take a temperature sensitive allele and you switch it off when there is the expression of gene. For example, in Drosophila, if in developmental biology, the way experiments that we plan up is to find it, to lose it, and to gain it. So you found it, it expresses in the eye disc. Now you have to lose it at the right time. So use a temperature sensitive allele to switch it off during that period when it was present in the eye disc. And then you go back to normal. So the gene is now happy, the animal is happy, and ask after its emergence what has happened to the developing eye. You get an answer. So you have a control not only on spatial level, but you have a control on the temporal level too. And that is important in Drosophila biology as well as it's a core of developmental biology. So if you look at Gilbert, you will see 3 things are important in developmental biology. Time is important, spatial relationship important, and third, above 2 are the most important. All right. So space and time, if you have a handle on it, you have a handle on your experiment in Dev Bio. Some of the genetic tools I'll rattle off. I have 5 minutes. All right. So the core of our genetic tool is a driver and US system. This is a binary system which was created by Andrea Brand and Perryman. It is, so a driver, what do I mean by a driver? Same thought that I was talking to you about, that if a gene expresses in eye, why should I shut it off from the whole animal? For example, in the last experiment that I told you with TS, temperature sensitive, when I say that I'm going to switch it off only during say larval time when it expresses in the eye disc, but I'm switching it off from all tissue irrespective of whether it is doing something or not. So gene X expresses it in eye and it expresses in brain. Suppose in that tiny window I have shut it off. I have shut it off not only from the eye, I have shut it off from the brain too. And then I'm celebrating, oh, I got a phenotype. Somebody can come and tell me, well, it is not a specific phenotype, it was a crosstalk between brain and eye. Since you shut it off in the eye too, you are getting a cumulative phenotype. How am I going to answer this? So I need a site-directed insult. All right. So this guy would be a promoter specific for the eye. And I have tagged up Galford, which we have stole from the East geneticist. And we have another line which has the upstream activator sequence, which is US. And we have tagged up our responder element to it. A responder element can be a reporter, can be an insult in the form of overexpression or a loss of function. Now when you cross these 2 lines, in the F1 generation you have a fly where the driver, that is eye specific, is driving the gene of interest, either up regulation or down regulation of it. In that way you are not insulting rest of the tissues, you are just insulting the tissue of your interest. So this kind of Galford US activity helps you to not only control the insult, it is also helping you to control the site of insult. With this guy in mind, people have designed several layers of complex genetics. One is the FLP-FRT cloning system. What is that? So we know that MyEX mutation creates lethality. It creates lethality even when I do a pulse. So does that mean I should abandon my interest? No. I should follow it up and should know what X is actually required for. So what is the solution? The solution is, this is a imaginal disk. Let's make our life simple. This is a structure or a primordia from which the wing will emerge. And what I'll do is, I will create patches of cells which are negative for this gene in otherwise normal scenario. And then assay what is the defect in these patches. This tiny little insult will never become a hindrance for the emergence of the fly, right? Again, it is very simple. You have the actin promoter or any kind of a ubiquitous promoter tagged up with your Galford, but it's not directly under the control of the actin promoter. You have spaced them out with a terminator sequence. You cross it with another fly, which will be a source of flippase. This flippase will elicit site-directed recombination, which will remove one of the FRT and also the termination sequence, bring the actin promoter juxtaposing it next door to your Galford. Now you have a constitutively active Galford present in those cells which have successfully removed the terminator sequence. All right. Now you have the Galford, and as I've told you in previous slide, you can combine it with any responder. You can combine it with a reporter line, thus visualizing the patches of negative cells. You can cell sort them to look at their size, their DNA content, analyze their clone size, so therefore you can comment on their cell division, or else you can overexpress any factor or take away the function of any gene. In a way, that otherwise seemingly normal background, you have patches of homozygous clone, giving you a total understanding of how this mutant is required, how the normal function of this gene is required during development. The tool that Subramaniam has described to you, lineage tracing tool, enables to lineage trace the entire animal in C. elegans. But we have used this to twist it around and ask question about lineage of particular cell type. Suppose you look at this particular cell and it seems to be very interesting and you want to know what is the fate of this cell. We have designed our cassette in such a way that, again, it's a Galford based, that you can permanently tag a cell and forget for lifetime now. Come back after 3-4 days when they are about to pupate and ask what structure have they formed. You would not only know the structure of this that they have formed, you would also know the other cells that have emerged from it and contributed in the formation of a structure, right? As you can guess that I am indicating something of stem cell biology, where we often use this tool which allows us to follow cell-fate decision of stem and progenitor cell population and their dissidence within a living organism, is based on the visualization of a cell lineage via time and spatially controlled reporter gene expression in stem cell progenitor cell that can be directly passed on to their progeny, and it enables us to study the dynamic of proliferation, differentiation, migration of the cells in an in vivo context during development, homeostasis and disease. So suppose you know that in a cancerous condition, in a cancerous condition, a particular gene expression changes, right? And you really want to find out that this gene was working at the level of progenitor cells or at the level of stem cells, right? What you need to do is to mark those cells which were expressing this particular gene, mark them permanently, so that the other, during mitosis, the other cells that come up from this cell, their lineage also gets permanently marked, right? So in a way, in a party, okay, better example would be a railway station in which a group of school kids have come over to take a train and the teacher suddenly lost track of the children. How do you identify them? By the school dress, right? In a cancerous cell, the cell positional information get lost. They are in all possible direction. Why? Because of metastasis. But how do you know that these cells once upon a time needed to be together to prevent this cancer formation? You would tag them. You would put a uniform onto them, right? And in a cancer situation, you would see all of them dispersed, means their normal adhesion has been objected. If you don't see that happening, you know that gene was not perturbed or not cause of cancer. But if you see a cancer cell has now dismantled its architecture and moved around in all possible direction, you have a result with you. I would end my presentation with a comment from one of our biggest scientists of our field, Jerry Rubin. He says there is a lack of appreciation for basic research in general, although basic science brings the fundamental breakthrough that underlies translational research. And I hope I have convinced you all that Drosophila can help you in achieving this. These are the few places where I actually had to jump in to get all these materials. Andreas Prokop and his group, Drosophila for School and Manchester Fly Facility, and 2 beautiful movies which I would encourage you all to look at. And of course, all researchers who have contributed towards the growth of Drosophila research. Okay, I'm open up for questions.
Thank you, Lolitika. For students, I know you are probably the best student here. Since the young students do not have questions.
I'm youngest.
Yeah.
So, Lolitika, I had a curiosity for long. I never bothered to ask anybody. So, in Drosophila, you cannot have sequence-specific recombination is what I understand. Like, generally you don't go for it. So, driver lines, how were they generated in the first place? Just random?
Driver lines by itself. So you have to get the promoter of this gene and then you fuse it with the Galford lines.
Oh, so you
make a construct, yeah.
You need to have the enhancer
information with you. Yeah, you have to have a construct with you. You have the enhancer information with you, design that, put it with the Galford, and then you get a transgenic fly.
Then you introduce it, it gets integrated in the genome randomly?
It can get randomly, or there are ways in which you can cite it off. Okay. That you can do. But you know, generally when we make Galford line, we pick up very specific genes enhancer. Right. So therefore, even you know, if it is eyeless, it has to express in eye. If it is blood, then something which is blood specific, which ensures, but otherwise you can target it also.
But when you do that,
the way maybe I'll just tell you, the way we do it, the other way to address your question is there are P element, right? So we often design P element which also has a Galford. When it is hopping around, then you screen for the lines which are in the right integration, and then you have to stabilize that line.
Yeah.
That's one way of doing Galford if you want to.
Because you will also have context-dependent variation.
Absolutely, absolutely. Absolutely, absolutely. Lot of steps, lots of screening has to be done. But good part is, you know, we have stock centers who actually takes care of lot of it. Until you are working on a very novel gene or you have a very specific question, then you have to invest your time. Otherwise, there are Bloomington, VDRCs and Querto, name it, and researchers are nowadays being very generous. Whatever land they are creating, they are dumping into these centers.
Mouse researchers get infected by that gene.
Yes. He's something very good of Drosophila biology. Yeah.
So, like, how is it that can you make every gene sensitive?
Yes, you can. It's already existing, lot of it. But you really want to make one, you can, because these TS alleles are always easy to incorporate with your gene action. What I didn't talk about is, we also have stolen Galati from East. And you know Galati can shut up Galford expression, right? So what we have done is, we have created situations in which we have a Galford there, and with the responder we have put a Galati. Now wherever Galati, and a TS element attached to your Galati. So whenever Galati is high, your Galford is down. So you are regulating the expression of Galford. So not necessarily always you have to create a TS, you can use Galati also. Yes?
I have one question related to like connecting Drosophila and aging. So we know like every developmental process, for example, first instar to second instar, second instar to third instar, and then pupation, and then adult life. These all have like fixed duration. So where
Aging actually in adult. Aging is addressed in adult, because all that you see in larvae is not really a mature thing. You are seeing different stages of development, which is good to understand a developing process, but not aging. Do you see? Aging you can only count when you have reached a perfect balance.
I was like trying to extrapolate first, like these.
We do have delayed larval period, but I don't think you can call it aging. You don't call it, because in that delayed larval period, everything is delayed.
Can't aging also be like considered as like it's an ongoing development process?
We call it slow development, but you see aging, our interpretation would be aging would be you are mature, you're mature adult, and then from 45 days of Drosophila life that you see, that is the aging, slow process of dying down, right? That we try to map. But having said so, also there are certain mutants which have emerged from this aging analysis that show kind of a epidermis phenotype. And some of them have now started being analyzed in late larvae and pupae too, that the onset is somewhat precocious, right? So people are trying to model the skin, the normal cuticular distribution, proteins and the lipids, how they change with aging. Is it precocious, is it expedited, or how is it?
The lifespan of Drosophila is 52 days.
52 days, well, not everybody. How would you consider the whole population? Whole lab population, 45 days.
45 days.
Without infection.
We know of like any mutants where lifespan has been significantly elongated.
There are. These mutants, you can look at, Chico mutants and others, they are elongated. I can talk to you.
Like, there is one gene, alpha-ketoglutarate, that has been associated.
But you see, alpha-ketoglutarate should play a role in multiple processes. So you have to pick a gene which is otherwise dedicated for, uniquely dedicated for aging process. All right.
Okay, there are no more questions. Let's thank Lolitika for this.
Thank you.